In this activity, we're going to compare different types of wood using microscope images of three common genera: Acer, or maple, Quercus, or oak, and Pinus, or pine. The goal is to learn how to recognize ring porous, diffuse porous, and non-porous wood based on what you see in cross-section. Wood, or secondary xylem, is not just one kind of cell. It's made up of several cell types, including vessels, tracheids, fibers, sclerids, and parenchyma cells. All types of wood also contain rays, which are bands of parenchyma cells that run sideways across the stem and allow lateral transport. Different tree species don't build their wood in exactly the same way. The size and distribution of vessels can vary depending on the species and even the time of year the wood was formed. By looking at how big the vessels are, how they're arranged, and whether vessels are present at all, you can identify different wood types. In ring porous wood, the vessels formed in the spring are very large, while those formed later in the summer are much smaller. This creates a clear, visible ring pattern in each growth ring. Many ring porous trees, like oaks and ashes, live in seasonal climates where spring growth is a big, sudden push. The trade-off is that those big spring vessels are efficient but more vulnerable to air bubbles and freezing damage. That's one reason these trees tend to reset each year with a new ring of large vessels. In diffuse porous wood, like maple, the vessels are more evenly sized throughout the entire growth ring. Instead of having a burst of huge vessels in spring and tiny ones later, these trees spread out their water transport more consistently across the growing season. That's why you don't see a dramatic contrast between spring wood and summer wood vessels in diffuse porous species. This strategy works well for trees that grow in environments where conditions are more stable or less extreme during the growing season. Water transport is steady rather than concentrated into one big seasonal push. In non-porous wood like pine, there are no vessels at all. Instead, all water transport happens through tracheids, which are narrower and more uniform than vessels. This makes water movement a bit less efficient than in vessel-bearing woods, but it's also safer and more resistant to freezing and air blockages. This system is typical of conifers like pines, spruces, and firs, which often grow in colder or drier environments. Using tracheids only is a more conservative and reliable strategy for moving water under stressful conditions. When people hear the words hardwood and softwood, they usually think it means one wood is hard and the other is soft. But in botany, those terms actually describe what kind of plant the wood comes from, not how hard it is. Hardwoods come from angiosperms, which are flowering plants like oak and maple. These woods usually have vessels or are porous, which you've been looking for in the ring porous and diffuse porous samples. Softwoods come from gymnosperms or conifers like pine, spruce, and fir. These woods don't have vessels or are non-porous and rely only on tracheids for water transport. So a hardwood isn't always hard and a softwood isn't always soft. The terms are really about plant ancestry and anatomy, not the physical feel of the wood. Before we get started with the activity, answer this question. Why do different tree species have distinct wood types like ring porous, diffuse porous, or non-porous, and how does this relate to their environment? As you think about this, consider how water availability, climate, and growth patterns might influence how a tree builds its xylem. Ask yourself why some trees might benefit from large vessels while others rely only on tracheids. Try to connect wood structure to environment and function. Before you try to decide what kind of wood you're looking at, let's walk through what structures you should always look for first. Start by looking for annual growth rings. These usually appear as concentric bands or repeating patterns that circle the stem cross-section. Each ring represents 1 year of growth. Within each growth ring, try to find the spring wood and summer wood. Spring wood is formed earlier in the growing season and usually looks lighter or more open because the cells are larger or have thinner walls. Summer wood forms later and usually looks darker or denser because the cells are smaller or have thicker walls. For diffuse porous wood, it is hard to distinguish between spring and summer wood. Next, look for vessels. If they're present, they'll appear as large, round, or oval openings in the wood. In some samples, you'll see many of them. In non-porous wood, you won't see any at all. Although in non-porous species like pine, you will see resin channels that often get mistaken for vessels. Resin, commonly called sap, is the thick, sticky substance that oozes out when a tree is damaged. Whether or not you see vessels, you should be able to find tracheids. These are narrower, elongated cells that make up much of the wood, especially in samples without vessels. You won't always see individual tracheids clearly, but they give the wood its overall fine, uniform texture. Finally, look for rays. These appear as lines or bands running from the center of the stem outward toward the bark, crossing the growth rings. Rays are made of parenchyma cells and are involved in lateral transport and storage. If you train yourself to find these features first, identifying the wood type becomes much more logical and much less like guessing. As you look at the images, you'll make sketches in your lab notebook and label these structures. Focus on overall patterns rather than tiny details. The goal is to show that you can recognize the key structures that define each wood type. Using what you observe, you'll decide which wood type each specimen represents: ring porous, diffuse porous, or non-porous. Use vessel presence, vessel size, and vessel distribution as your main clues. Answer this compare and contrast question. Explain how you distinguish between the three wood types. As you answer this, think about what specific features you used. Did you focus on vessel size, vessel distribution, or the absence of vessels? Try to explain your reasoning using observable anatomical differences, not just the names of the categories. This activity shows you that wood is not the same in all trees. Its structure reflects both function and environment. Being able to recognize these patterns helps you understand how different trees move water, grow, and survive in different conditions. It also strengthens your ability to interpret real plant anatomy from microscope images.